All our immunology books are filled with antigen and antibody talks. But what if you just need an overview for antigen and antibody interactions? Do not worry and watch this video till the end. An antigen, by definition, stimulates the production of antibody, which in turns combine with the antigen. Both processes are based on complementarity or fit between two shapes. A small piece of antigen or determinant and the combining site of the antibody. A cleft is formed largely by the hypervariable regions of heavy and the light chains. The closer the fit between this site and the antigenic determinant, the stronger the non-covalent forces between them, and the affinity will be higher. When both combining sites can interact with the same antigen, for example, inside a cell or pathogen, the bond has a greatly increased strength, which is termed as avidity. The ability of a particular antibody to combine with one determinant rather than another is referred to as specificity. Well, if it's combined with another antibody, it's not going to react. The antibody repertoire of an animal stored in its V genes and further explained by mutation is expressed as the number of different shapes toward which a complementary specific antibody molecule can be made, and the copies are run into millions. Then, what happens when antigen and antibody combines? It completely depends on the circumstances. Sometimes antibody alone is not enough to neutralize the antigen. This is the case for toxins such as tetanus or diphtheria or certain viruses. They need to attack the cell surface receptors to gain the entry, and the process is termed as neutralization. You can find the process of neutralization in one of our scan-animations on sqadia.com. Well, now let's get back to the topic. In the secondary interaction of the antibody molecule with another effector agent, such as complementary or phagocytic cells, is required to dispose of the antigen. The importance of these secondary interactions is shown by the fact that deficiency of complement or myeloid cells can be almost as serious as deficiency of antibody itself. The combination of antigen and antibody is termed as immune complex. The immune complex can be small and soluble or may be large but precipitating, depending on the nature and proportions of antigen and antibody. The usual fate of these complexes is to be removed by phagocytic cells via phagocytosis, through the interaction of the Fc portion of the antibody with complement and with cell surface receptors. Another fate that it follows is that complex may persist in the circulation and cause inflammatory damage to the organs or inhibit useful immunity, for example, in the case of tumors or parasites. So, that was all about the intro of antigen-antibody interactions and immune complexes. Let us get into the main topic, so keep watching sqadia.com.